So our first group of organisms to consider are the prokaryotes. So what is a prokaryote? A prokaryote is an organism that lacks a nucleus. And that means a lot of other things about a cell if it lacks a nucleus. Now, taxonomic clades, as we just talked about with systematics, are best defined using not shared ancestral characteristics, simplesiomorphies, or unshared derived characteristics, which are autapomorphies, but by using synapomorphies, or shared derived characteristics. So lacking a nucleus, we know that eukaryotes are considered to be more advanced. Having a nucleus is a derived characteristic, which means that it's opposite. Lacking a nucleus is not a derived characteristic. It's not an apomorphy. It's a plesiomorphy that's shared by the two domains of prokaryotes. So it is a simplesiomorphy. Lacking a nucleus is a simplesiomorphy. Therefore, prokaryotes that share this ancestral condition are not recognized as a biological taxon. They are not a clade that contains all of the living members of a group of organisms that shares common ancestry. So we should really describe prokaryotes with a lowercase p to indicate that it's not a taxon. But here it's at the beginning of the sentence, so we know that it should be capitalized. So we have two domains of, here we can see lowercase p prokaryotes, bacteria and archaea, which used to be lumped together as one kingdom called Monera, M-O-N-E-R-A. That's an old name, so you don't really need to know it. But using biochemical techniques, we had to use the morphological differences to distinguish between different lineages of prokaryotes. And then Dr. Carl Woese, who was a microbiologist, passed away fairly recently, back in 2012, looking at the sequence of nucleotides in a particular gene, determined that bacteria and archaea were in fact two different clades of single-celled, nucleus-lacking organisms. So he proposed that there should be a separate domain called archaea, and he based that proposition on the sequences of genes encoding ribosomal RNA, the DNA sequences for those genes. So he looked at the nucleotide sequences, lined them all up, and saw where were there differences, where were there similarities, and determined that, lo and behold, they were not all one big happy family, but they were actually two clades that were very distinct from each other. And why did he use this ribosomal RNA gene? Because all organisms have it. And because all organisms have it, you can make comparisons across very different groups of organisms. That's one benefit. And because it is an essential gene, every organism has to have it and has to transcribe and translate it pretty much constantly, the rate of mutation is very slow. So you make a mutation in ribosomal RNA genes, and chances are bad things will happen unless those mutations are very slight. They make very slight differences, very small changes, so their rate of evolution is very slow. So good for comparing very broad groups, because if the genes might mutate too quickly, you can get a lot of homoplasy, and that's no good. So he proposed splitting out the archaea, or a large group of organisms that he hypothesized were ancient, as the domain archaea, sometimes called the archaebacteria, and they were hypothesized to be very ancient because they were found in habitats that most scientists thought were reminiscent of what the ancient Earth was like before we had an oxygen-rich atmosphere. The Earth went through what's called a Hadean period, where there was a lot of volcanic activity, no oxygen in the atmosphere. And a lot of these archaeans are found in these types of habitats where there's no oxygen gas around, very hot temperatures, extremes of pH and salt. However, looking at the data and trying to line up where the archaea fit within the Great Tree of Life, the data tell us a different story. They suggest that archaea are actually a sister group to the eukarya. So here are the eukaryotes, here are the archaea, and here are the bacteria, the true bacteria. So another way to say that prokaryotes are not a good taxon is to say, if we look at this like it were a tree, and we were pruning the tree, there's not a good way to cut out from the common ancestor a prokaryote clade that includes all descendants from that single common ancestor, including all those organisms and only those organisms. So if we were to cut down, snip out all of our prokaryotes, we'd have to make two cuts. We'd have to cut here by the common ancestor, but then we'd also have to cut over here to eliminate the eukaryotes. And since we can't do that, we can't rearrange the topology of this tree, the shape of this tree, to make a group, a natural group, that includes just bacteria and archaea without also including the eukarya. So in spite of the morphological and the physiological evidence, archaea and eukarya form a monophyletic clade that is distinct from the bacteria. It also suggests that the bacteria are an even more ancient lineage than the archaea, but there are ways that that could not be true. And I'm not going to go into those right now, but what we can say definitively is that bacteria and archaea are not necessarily sister to each other, because eukarya and archaea are sister to each other, and the archaea-eukarya clade is sister to bacteria. So in addition to not having nuclei, there are a number of other symplesiomorphies that are found within the groups that we consider to be prokaryotes. They don't have the nuclei, of course, and that means they don't have other membrane-bound organelles as well. So you're not going to find things like Golgi apparatus. You're not going to find endoplasmic reticulum. There are a lot of those organelles that we find typical of eukaryotic cells. Prokaryotes are also unicellular, single-celled organisms, or colonial, meaning comprised of a bunch of similar type of cell, not a lot of differentiation, no differentiation actually. And if we remove one or a few of those cells, they can grow into a new colonial organism. So that's the difference between being colonial and being truly multicellular. The cells of prokaryotes tend to be very small, on the order of 1 to 5 microns. That's millionths of a meter. Eukaryotic cells tend to be on the order of 10 to 100 microns in size, although there are exceptions to this rule of prokaryotic cells being very small. And the morphology, or the shape of the cells in prokaryotes, is fairly limited. Not a whole lot of different shapes like we see in eukaryotic cells. So by and large, the cells of prokaryotes come in 3 shapes. They can be little dots or really spheres or ball-like structures called cocci. If you just have one, it's a coccus. They can be rod-shaped or kind of look like a capsule, like you might take if you are trying to consume some medicine. Capsule-shaped or hot dog-shaped if they're kind of like a long capsule. Those are called bacilli. Just one, you've got a bacillus. Or they can be kind of corkscrew-shaped, spiral-shaped. And that's called a spirillum. And typically there's many of them, so we call them spirilla. So here, for example, is an artificially colored electron micrograph of Staphylococcus aureus, which is a coccoid or sphere-shaped cellular morphology. You can see they're very, very small, less than a micron, about half a micron in diameter. This is another artificially colored electron micrograph. This is E. coli, which is a bacilliform or rod-shaped bacterium. Again, they kind of look like hot dogs. And then this is Campylobacter jejuni, which is a pathogenic fungus typically associated with things like uncooked meat. And you can see it's kind of somewhat corkscrew shaped. So these are spirilliform or spirilla bacteria. You can see they've got some appendages on them as well. We'll talk about those appendages too. So sometimes you may have heard of something like Staphylococcus. Here we've got Staphylococcus again. Staphylo is a Greek prefix that means cluster of grapes. And we can find colonies of bacteria that look like they are attached to each other, kind of like a cluster of grapes. So the name Staphylococcus is really descriptive. It describes the morphology of the colonies under the microscope. And if you ever had a staph infection, that is an STAPH infection, not an STAFF infection. And that's short for Staphylococcus. Now, strepto is another prefix, and that comes from the Greek word meaning twisted chain. Or I usually just think of it as like a chain of cells. And here you can see Streptococcus, little spheres, spherical bacteria that are arranged in a chain. And Streptococcus, one species of Streptococcus, is the pathogen, the disease-causing agent for strep throat. So if you ever had strep throat, that's where that prefix strep comes from, which refers to the morphology of the colonies when you look at them under the microscope. So how can we distinguish between different types of prokaryotes? One quick and easy way, relatively quick and easy way, is to look at their cell walls, the chemistry of their cell walls, using a technique called gram staining. Gram staining was developed by Hans Christian Gram, who is a Danish microbiologist. And he found that with this stain that he developed, the staining technique, certain types of bacteria would show up as dark purple. You can see them in here. Some dark purple gram-positive bacteria. Others would not absorb the stain, and they would turn just kind of pinkish, and those were called gram-negative bacteria. So a gram stain is a diagnostic device to examine cell wall composition. And so what's happening is because prokaryotes typically have cell walls, the cell walls are made up of a substance that's different from eukaryotic cell walls called peptidoglycan. Peptide, you might think that sounds like protein or amino acids being linked together, and you'd be correct.